thank Mohammed and all of the organizing team for putting together another great lineup. I've been lucky enough to attend pretty much all, not all, the oligo conference, and it's nice to see it going from strength to strength. And the list of speakers today is a very good show of strength of this field of oligos. I do share quite a lot in common with Margot, actually. I think we did. I also come from a nice part of the world where we have nice sea, the mountains, good wine, good strikes. He beats me by the number of years he spent in Cambridge. I only spent 5 years, he spent 7. I do beat him in terms of number of gray hair, so we're very safe. So I'm just going to present a little bit of the work we're doing in my group, which focuses mainly on the engineering of platform technologies for the detection of cell-free nucleic acid biomarkers in a broad range of bodily fluids, which we call in here liquid biopsy. And then we try to develop, I'm a chemist by training as well, so we try to develop molecular probes, develop technologies, and then try to implement them within devices that can be used in clinic, either at the point of care or at the point of need, in order to replace the more invasive, expensive, and time-consuming tissue biopsies. So the type of biomarker we're particularly interested in my group are circulating cell-free microRNAs. I'm not going to go for this audience into the details of what microRNAs are. Very briefly, short non-coding RNAs, 20-25 nucleotides in length, that just play key roles in the regulation of gene expression by binding to messenger RNAs. So they play this key function within cells, but they also tend to end up in circulation in pretty much all bodily fluids, blood being the most common, urine, saliva, and also skin interstitial fluid. And they do so by a range of different mechanisms, some being more active, using some transport protein or extracellular vesicles, some more passive, when cell dies, basically releasing the cargo, and then some of them ending up into circulation. So they are a great source of information, but also a great source of biomarkers. They have pros and cons. This is true for pretty much all biomarkers. The pros, they can be sampled in a minimally or non-invasive way, depending on which fluid you are medically interested in. They have great diagnostic, prognostic, or predictive values, and so for a broad range of disease and pathologies. A lot of the work we do in my lab is trying to use these biomarkers for early cancer diagnosis. Today, I'm going to talk about something very different, but still using the same type of biomarkers. They tend to have a high level of sensitivity and specificity, mostly when you look at a panel of different microRNAs, some being upregulated, some being downregulated. You have very limited information when you look at the single microRNAs, but you have to look at 2, 3, 4, and sometimes much larger number of microRNAs. So those are the pros. The cons, they tend to be present in systemic circulation at very, very low concentration because they are mostly diluted with microRNAs or other metabolites coming from anywhere in the body. There's hundreds of microRNAs known so far, with 20-25 nucleotides long. So obviously, by definition, they will have a very high sequence homology. Some microRNAs differing with another by 1 or 2 nucleotides. And the last cons, especially for diagnostic or sensing application, it's very difficult to detect microRNAs directly from bodily fluid. You will normally need to do some heavy sample processing, extracting the microRNA, amplifying it in some cases before you can actually detect them in a quantitative manner. So as I said, most of the work we do in the lab is focusing on cancer diagnostics. I decided today to present something very different, and which is trying to use microRNA to develop a predictive blood test for prediction of preterm birth from as early as 12th week of pregnancy. So why focusing on preterm birth? Why is it important to detect it or predict it early in order to prevent this event? So first, what do we mean by preterm birth? It's basically any birth that will occur before the 37th week of pregnancy. Very, very common, sadly, with 15 million preterm births happening every year, leading to 1 million deaths. And that means around 10% of babies are actually born in the world prematurely. So quite not something so rare, something that can happen, sometimes with no consequences, but sometimes with quite heavy morbidities. The sad thing in here to the story is that more than 3/4 of preterm birth could actually be prevented, especially if they can be predicted. So if you know women are at risk of delivering preterm, then there are ways actually to preventing it from happening if you actually kind of monitor them more closely. So why is it so difficult to kind of prevent or detect preterm birth? Is because preterm birth can have many different causes. So many causes, different factors, it's very hard to actually kind of know which woman is likely to kind of give birth preterm or not. For example, it occurs very, very commonly in young women not classified at risk who are having their first pregnancy. Very healthy first pregnancy, so no kind of previous history of preterm birth. So you wouldn't define them as being at risk. And the current diagnosis of preterm birth is highly inefficient and can only predict imminent birth. That means that you can have a test, it will tell you whether you're likely to deliver preterm in the next kind of 2 weeks or so, which is not kind of very kind of give you enough time to intervene. The positive side of the story is there are ways of preventing preterm birth. You can have chemical treatment or more kind of physical intervention, but will be kind of available to clinicians, but will be able to prevent women from delivering preterm. That can be from kind of injection of progesterone, for example, or kind of more kind of invasive kind of cervical circlage. But to be able to do that, for this treatment to be effective, you need to be able to do this early enough in the pregnancy to stop the kind of preterm labor. As I said, the two most common kind of techniques for kind of predicting preterm birth are cervical length measurement, which is normally done within the second trimester of pregnancy. Again, quite expensive, doesn't give you much time to then to intervene, and is not being done as a routine testing for all women being pregnant. The other one, which is more kind of a biochemical test, looking at fetal fibronectin, which is quite simple, non-invasive, looking at the blood test, but can only be used after 20 weeks of pregnancy, so when you're already kind of quite far into the pregnancy, and is not useful for distant prediction. So this is where we come and trying to see if we can kind of develop a new test, which is going to be based on microRNA biomarkers, to try to kind of have a better risk stratification of women early on in pregnancy. So for example, when women have their first prenatal kind of appointment, around 10 to 12 weeks of pregnancy, where they actually have to kind of have the appointment anyway, can we have a new blood test that will allow you to kind of classify whether they are at risk of delivering preterm and then being monitored more closely throughout the rest of the pregnancy, or thinking, no, they're fine, they can carry on as normal, the risk is minimal. So what do you need to do that? Well, you need two things. You need some highly specific biomarkers or combination of biomarkers. But you know the answer already. I've mentioned it before. So we're going to look in here at microRNAs. We were lucky enough when we started this project to collaborate with a team of clinicians at Imperial College London, led by kind of Dr. Basso Terzidu, who had identified in maternal blood as part of a kind of clinical trial a batch of microRNAs that were shown to be deregulated early on in pregnancy. I will come back to that in the next slide. But we also need then, when you have a biomarker, you need some minimally invasive tests that are affordable, robust, and easy to use, so that you can try and detect these biomarkers in a kind of a very kind of large scale kind of a screening fashion. So as I mentioned, starting point from a clinical collaborator was this kind of a clinical trial, which was done on high-risk population of women kind of likely to deliver preterm. They've collected samples at 3 different time points for pregnancy. The first one being kind of the 10 to 12 week of pregnancy, then 14 to 16 or 20 to 24, and then analyze all microRNAs at all of the different time points using kind of encounter nanostring technology at the initial kind of detection methods, and then RT-qPCR to validate the biomarkers we have identified so far. This is too small, you can see it, but that's not the point anyway, but just to show you a lot of data that they've done, and they've identified quite a few different microRNAs. The best candidate out of the study were MIR155P, which had an area under the curve as a single microRNA of 0.86, with a 4.54 decrease in expression compared to kind of women delivering a term when sampled between the 12 and 14 week of pregnancy. The second one was MIR 374A5E, which by itself had an area under the curve above 0.8. So in the rest of the talk, I'm going to mention kind of technologies which have been targeting these two different microRNAs. So again, don't have to kind of spend too much time on that. There are technologies out there to detect microRNAs. Obviously, PCR or RT-qPCR in this case being the gold standard. Works pretty well, especially at the lab-based techniques, but have quite a few kind of downside. Amplification-based, you use enzyme, there's two amplification steps, but the reverse transcriptase plus the PCR of a kind of complementary DNA which has been generated. They all tend to be prone to contamination and sources of error. This is particularly true for small RNA, like the kind of microRNAs, even more complicated than trying to do RT-PCR for longer RNAs. And there's also, as I mentioned before, a requirement for sample processing. You actually have to extract the nucleic acid from the samples before you can actually do any RT-PCR. All of this meaning great tool for lab-based, not a great tool for kind of a diagnostic kind of a device that can be miniaturized and rolled out to large population. This is why kind of chemist engineering, whatever you define, I call myself depending on the audience, we try to kind of develop an alternative technologies to kind of RT-qPCR, but be more amenable to incorporation into small devices. And we've kind of worked for quite a few years now on the strategy based on the concept of oligonucleotide templated reaction, where we basically use the microRNA target as a template to catalyze a fluorogenic reaction. So that's the strategy represented in here. The target microRNA is shown in red. We are able to develop two molecular probes, one in blue, one in green, which are both chemically modified with kind of gray boxes there, which are kind of chemical groups which are non-fluorescent, but can react with each other to form a fluorescent product. So if you put all of this together in solution, there's no fluorescence being generated. When the microRNA is present, when one probe binds to the 5' end of the microRNA, the other one to the 3' end. By doing so, it brings close to each other these two molecular groups that can then chemically react with each other to form the fluorescent kind of dye. That means for every microRNA target present, you will have one molecule of fluorescent dye being produced, and you can have a kind of sequence-specific and quantitative kind of detection of the target. So the scaffold we use for our probes are peptide nucleic acids. I'm always surprised when I come to this conference how DNAs are not so popular. I guess we hear always a lot about kind of LNAs or kind of any kind of chemical modification of the sugar backbone. DNAs not so much. Can understand that for anything which is therapeutic application. For diagnostic or sensing, DNAs have many advantages. Easy to synthesize, chemically modified, scalar, peptide synthesis. So kind of great kind of advantages, at least for sensing. We don't want them to get into cells, so no problem with that either. So how do we design them? That's the example here on the bottom. That's the microRNA sequence. We basically highlighted in red the two sequences we're going to target with our DNAs. DNAs tend to be 7 monomer long, which is just enough for them to hybridize to the target at room temperature. They're not very water soluble, people know. Neutral backbone, so to try them to make them water soluble, we add 2 arginines at kind of either end of the DNAs, and then chemically modify them with the molecular probes so that we can actually do the sensing. For the sake of time, I will keep a long story short. This is the chemistry we're using, shown here on the left. One DNA is modified with a kind of a quenched coumarin, the other one with a thiol, and you have a 1,4 addition, is basically unleashing the fluorescence of the coumarin. We've done all the traditional in vitro testing with targeting DNA to show it was quantitative, it was fast, it was sequence specific. Because there's no amplification, you can use kind of a contamination from heparin. If you collect your blood in a heparin tube, for example, there's no issue having to degrade the heparin treatment beforehand. So just going to quickly show that. Works very well in solution. How do you go from something that works in solution to a device that can be used in clinic? Ideally in the bedside, and then at the point, I tried to kind of stretch that this was well before COVID, when people were using kind of lateral flow tests kind of twice a day. That's kind of what the idea, can we actually use the paper-based and implement our OTR sensing onto paper? How do we do that? So again, you show on the top the general strategy. We have 2 DNA probes. We have one which is a capture probe, which we're going to kind of the one modified with the file. We introduce the biotin at the other end of the PNA, which contains the file, bind it to polyscriptavidin, print this onto the test line of the lateral flow test. This is the one in green there with the kind of the pink ball representing the biotin. The other kind of sensing probe is the one that contains the coumarin. This is the one that is going to be flowing alongside the lateral flow. We print it just under the sample loading pad, where you're going to deposit your clinical sample. When you add your sample, that will be blood or plasma in this case, is going to be absorbed onto the sample loading pad. If it has microRNA complementary to the kind of sensing probe, going to hybridize, then weak alongside the LFA. When it reaches the kind of test line, will be captured by the other probe, and then the same reaction of OTR will take place exactly on this position, and your test line is going to kind of start lighting up. You now concentrated on a very small area of your test, all of the fluorescence. Any background fluorescence coming from the unquenched probe is now kind of washed off at the other end of the lateral flow test. And you end up with no RNA, the probe non-fluorescent, with the RNA present, the signal of fluorescence, period. How do you analyze your test? Well, initially, to make sure we could visualize it, we use the fluorescence scanner, few 100,000 pounds. Don't really expect kind of clinics to have that in the kind of in their office, but allows you to visualize the band here at the fluorescent band. You can also use a much kind of cheaper, a few thousand kind of pounds, kind of a benchtop reader, which is the one on the top, commercially kind of sold by QIAGEN. You can just put your device in it, and you will be able to see a trace, like in the bottom, almost like an HPLC trace, with a fluorescence reading, and you can measure the intensity of a peak. And we have pretty similar limit of detection with these two instruments. Again, we've done the same general strategy. Those response to make sure it was quantitative, sequence specificity, down to single nucleotide, introducing mutation in different areas of the microRNA target. We were able to achieve a limit of detection down to femtomolar concentration and the specificity down to single nucleotide mutation, again, depending on the position of what your microRNA target. So always happy with that. How do we move then to kind of preclinical validation? Benefit of working with clinical collaborators. We actually did with biomarker discovery studies. We still have some samples available. So we basically kind of try to take some of the samples from them, did the RNA extraction, and then use 5 microliter of this microRNA extract onto our lateral flow test. And we were able to kind of see some statistically significance between the women we ended up delivering at term and those we ended up delivering preterm. So it was statistically significant, which was great and very promising, but you still needed to do the RNA extraction. So then it just takes away the purpose of doing a point of care test, if you still need to kind of spend a few hours and then kind of kick in and out to extract the RNA from the sample. So we tried, okay, let's be bold, can we actually kind of load directly some plasma onto our lateral flow test, and can we still detect something? You don't kind of hold much hope for this experiment, but let's try it. And to our surprise, we still be able to detect some signal and some kind of weak statistical significance difference between the term and the preterm group. So yeah, it was promising, but barely statistically significant, so not enough to build a proper diagnostic test. But in the end, we were kind of pretty much kind of surprised of kind of, well, not surprised, but it was kind of so weakly different because microRNAs in blood will tend to be kind of rarely freely circulating, they either trapped within exosomes or bound to proteins. So the question was, why did it even work in the first place? Because if it's just all in exosomes or in proteins, why can you actually detect some kind of signal here? Well, then we started kind of trying to look at what we have in our running buffer for the LFA, and the buffers contain some kind of twin, which is normally there to regulate the flow onto the paper when the actual samples kind of whisk along. And if you look into more detail and we did the experiment, we actually need the twins to have a signal detected, because the twin is actually able to kind of denature the exosomes and release at least the exosomal fraction of the microRNA we want to detect. And near 150, which is the one we're detecting in this experiment, is known to be mostly exosomal as well. So that's probably why we could actually detect something in the first place there. But twin is not kind of a strong enough denaturing agent to kind of denature protein, our protein, for example, and release those kind of sort of microRNAs. So can we go further and kind of expand the pool of microRNA we can detect? So we try to increase and add a stronger denaturant, in this case SDF. SDF, we showed, we didn't reinvent the wheel there, but we just wanted to show it in the lab, can denature protein at kind of millimolar concentration and can also break down kind of extracellular vesicle like we exposed. So then we try to see, can we actually use SDF in our LFA test to try to kind of to optimize the detection. We didn't want SDS in our running buffer because SDS can potentially mess up with the detection part. So can you actually include SDS in our sample loading pad to have some kind of in situ kind of processing of a blood sample? And we've done that, again, keeping a long story short, we've kind of modified our sample loading pad, which is here on the left, highlighted by the red arrow, which is normally a big kind of a paper-based pad. We now kind of impregnated this kind of cellulose with an algidate hydrogel, cross-linked with calcium, and also containing ASDS. So you now have this kind of hybrid material containing cellulose and hydrogel, so it can actually absorb pretty well any samples you deposit on it, contain ASDS, denature proteins and exosomes, and should in theory release all microRNAs onto the LFA. And again, we tested that using only 3 microliters of blood plasma unprocessed onto the LFA, containing this new sample processing pad, and then we could see much stronger signal than without this pad in the first instance, and a much greater statistically significant difference between the two groups, term and preterm. And if you plot, again, with the caveat there, it's a pretty small number of samples, but we have an area under the curve of 0.97 here, which is similar to what we could get by RT-PCR analysis. So trying to expand that, but that was already pretty promising. There's some kind of conclusion on this part of the talk. We were kind of quite happy to have a lateral flow test that enabled the detection of MIR-150 from as little as 3 microliters of blood plasma. We've developed a new biomaterial for automated on-chip extraction. There's an amplification on-chip detection, and we are capable of predicting the true outcome of pregnancy at a very early stage from the 12th or 14th week of pregnancy. So that's the bright side of the story. There's always a kind of a darker side, because as I said before, to get an accurate diagnosis, you also need to kind of have more than 1 microRNA. You would say an easy lateral flow, you have multiple test lines, 1 for each microRNA, and that's going to kind of be easily solved. We've tried, and it does work to some degree, but not kind of as simple as we were hoping for. Some issues which we believe come from the fact that we're now printing PNAs onto lateral flow tests, PNAs being quite hydrophobic. So the flow to pass the first test line is okay, but then it stores at the PNA test line and then disrupts the flow for what's happening further down. So the test line beyond the first one are actually strongly affected by this kind of hydrophobic barrier in the first place. So we're working on it, but this is kind of proving more challenging. Also, limitation in terms of sensitivity. I think, as I mentioned, the technology is based on the principle that every molecule of microRNA generates 1 molecule of fluorophore. If you have a very poorly abundant microRNAs, you're going to be very quickly limited by how many fluorophore you can produce and then detect. So we believe that this is going to be a problem in the long run. And also the lateral flow test reader we're using is inexpensive, as I said, 3,000 or 4,000 pounds, and it's not a big ask to kind of have kind of obstetrician centers to invest in one. It's still something which is not readily available and will have to be introduced in clinic. So, having said that, while we continue working on the lateral flow test aspect of the project, we try to see if we can come up with alternative sensing strategies, which are now based on the sandwich assay. And then I'm moving into an area which is kind of much more recent and haven't been published just yet. So the strategy in here, we're still using PNAs, that's what we love to use in the lab, but we now are kind of trying to amplify the signal in a way by actually now not using 1 molecule of fluorophore per target, but using silica nanoparticles, the one on the right here in yellow, which is basically loaded by kind of thousands of different molecules of fluorophore attached to the silica nanoparticles. Those particles are then functionalized on the outer sphere by kind of epoxy group that can then be modified by a peptide nucleic acid through kind of the ring opening with the file, the cysteine group. On the other end, we're using kind of more commercially available kind of magnetic kind of microbeads, which are coated with streptavidin, which we can then functionalize with bioattenuated PNF, which will be done on a few more slides. The concept sandwich assay, as I said, you've got the microbead on the left, the silicon nanoparticles on the right. Microbeads about kind of 5 or 10 micrometer diameter, nanoparticles 100 nanometer diameter. But the microRNA kind of bridges the two, you know, end up with a magnetic bead, which is going to become fluorescent. And we've applied that to the detection of MIR3748, our second microRNAs. Detection methods, we're using now flow cytometry to detect it, which is an instrument which is commonly found in any kind of obstetric clinic clinics, because they're using it for many other things. And the principle is the following: if you have a mixture of magnetic beads non-functionalized, nanoparticles fluorescently kind of loaded, and the complex from the sandwich assay, where the magnetic beads can pass through the laser, they are non-fluorescent and won't generate any signal. The silicon nanoparticles can also pass, but they are too small for the limited detection size of a flow cytometer, so you won't detect them. You will only have a signal generated or an event created when the magnetic beads is functionalized with the nanoparticles to form a complex. We've done some, again, in vitro validation. Very happy to have a limit of detection of 70 attomolar for this technology, the strength of the kind of flow cytometer, a very good sequence specificity. 1, 2, and 3 here are free off-target microRNAs that don't seem to be generating any signal. So pretty happy with that. And to show that the complex is working, you have 2 TEM images on the left. The one further on the left is the microbeads without, the magnetic bead without the kind of microRNA present. The one on the right, you can see the smaller beads corresponding to the silicon nanoparticles, which are now kind of bound to the magnetic beads, where the microRNA forms a bridge between the two. And then we've been able to do, again, the preclinical validation. That's my last kind of result slide, to showing that you could actually have a pretty kind of clear, statistically significant difference between the term and preterm group using 5 microliter of unprocessed plasma directly with your sample. It can also work if you can change the color. You can replace the one that done with FITC nanoparticles. If you replace FITC by the CY5, you can also get the same, which obviously tells you that we should be able to do multiplex. One microRNA with FITC, the other one with CY5. I would have loved to show you that, but just kind of ongoing experiments. We've been done in clinic, blood taken during the first prenatal appointment. As I said, 10 to 12 weeks is normally when you will actually have your first appointment anywhere. Blood is already collected from women at this point. We would just need to take a few microliters of the plasma during a normal blood test to use it to scan through a flow cytometer and then hopefully do some kind of more effective kind of patient stratification. All right. And just to conclude, this is the work pretty much primarily of one PhD student who is now finishing, Lucia Petru, who kind of would have loved to be there, but was at the conference in Brisbane, so might be on the plane back. That's her with kangaroos. She sent me a few weeks ago when it was pouring down with rain in London, but that's all right. Our financial sponsor, the March of Time, and the clinical collaborator, as I mentioned, we did all of the biomarker discovery work. And thank you for your attention.